Bismillah ar-Rahman ar-Rahim. This is the new syllabus 2023, and we are now going to discuss chapter 5, which is on enzymes. And this is the first video on that. The normal routine is that we first look up the syllabus for the chapter 5, which is enzymes, enzyme action 5.1. Describe a catalyst as a substance that increases the rate of chemical reaction and is not changed by the reaction. So it keeps on being used again and again. The enzyme is not changed. Then describe enzymes as proteins that function as biological catalysts and are involved in all metabolic reactions whichever occur in the living organisms, whether it's plants or whether it's animals. Explain enzyme action with reference to the word substrate, active site, enzyme substrate complex, and product. So four words that we must know. So what is a substrate? What is the active site? What is an enzyme substrate complex? And what are products? Product or products. Explain the specificity of the enzyme. The specificity of the enzyme is the shape of the active site in terms of the complementary shape and the fit of the active site with the substrate. And then we have to mention the lock and key hypothesis, which needs to be discussed. And then we will discuss 5.2, which is effects of temperature and pH. Understand that the progress of enzyme catalyzed reaction can be followed by measuring the concentration of reactants and products. Investigate and describe the effect of temperature and pH on enzyme activity. So we need to do two practicals on this. And then explain the effect of changes in temperature and pH in terms of kinetic energy, shape and fit, denaturation, and the frequency of effective collisions. Now, that's a new word which has been used in this syllabus. It wasn't in the previous syllabus. Effective collisions. Now, this diagram is very well depicting the whole story of enzyme. So every enzyme has a depression on its surface, and this depression is called the active site. Not the rest of it, but just the place where the substrate is going to fit. That's called the active site. And the substrate molecules can be two or one molecule breaking up into two or two joining to form one. And this will be enzyme plus substrate, and this will form an enzyme substrate complex. Enzyme substrate complex. And then we have the enzyme, and then we have something else, and that is the product. So the product can be one, or we can say products. So but substrate, we don't use the word, we don't use the plural for it, we just say substrate could be one or two. So the substrate combines with the enzyme, and the enzymes are specific. Why? Because the shape of the active site is specific. So only one, a certain substrate which is complementary to the active site. The word is complementary. Please understand this word. The shape of the substrate is complementary to the shape of the active site, and that fits in it, and then the product is formed. But what happens is, because it is helping to form that bond, the enzyme is helping to bring these two together and then form the bond between them, which then makes it into one product. The enzyme, and the key is the substrate. So here we have the enzyme, which is the lock, and the substrate is the key, and then it forms the lock key complex. But there is no such thing in biology, but it is called the enzyme substrate complex. So the lock is always the enzyme, and the substrate is always the key. So this has to be very clear to you. In any experiment in which say starch, if I give you the famous one which we use a lot, starch is converted to maltose, and the enzyme is amylase. So amylase is the lock. And what is starch? Starch is the substrate. And starch is the substrate, so that is the key. So understand that the lock is the enzyme and the substrate is the key which actually fits into it. And then of course the substrate will either join, or it will form one molecule, or it will break up into two molecules. Substrate spelling should be correct. Enzyme, enzyme substrate complex, enzyme product. Here we have two things, so it's called products. Another diagram showing you how bonds break under stress when the enzyme substrate complex forms, and then the products are two separate molecules, and the active site, and then the enzyme. And here another one, we have two substrates joining, and it forms one product. So of course it is going to help to join these two separate 1 and 2 together. This is 1 and this is 2. So we join it together and we get the product. But the enzyme is always working and enzyme will always be available. It does not take part in the reaction. Now, I want you to understand this, though it's not in the syllabus, activation energy. You see, the reactants need to have a certain energy, and this energy is called activation energy. And then you see the reaction occurs and the product is formed. So this is the activation energy, right? Now we say enzymes lower the activation energy, so less energy will be needed for this reactants to form the products. So progress of a reaction with enzyme, progress of a reaction without enzyme. I just want you to know this. You can't get this graph in the exam, but we will do it in the A levels if you study A levels. So this is the fact about the activation energy, but I just want you to know about it. Enzymes are protein in nature and they're biological catalysts. Biological means when you study biology, you study living organisms, bacteria, animal, plants, fungus, lizard, mushrooms. So all these are biological catalysts found in living organisms. And they're specific by the shape of the active site is complementary to the substrate. And they're denatured at temperatures above their optimum. Every enzyme has its own optimum. So it will be denatured at temperatures above the optimum of that enzyme. If the optimum of that enzyme is 37, it will be denatured at 50. If the optimum of that enzyme is 90, it will be probably denatured at 110. And they work best at an optimum pH and an optimum temperature. Then what are biological catalysts and chemical catalysts? If biological catalysts are boiled and added to the substrate, then no products will be formed. That means no reaction will take place. If a chemical catalyst is boiled, it will still work and the reaction will take place. So there's a difference between biological catalyst and a chemical catalyst. Then without enzyme, why do we need enzymes? You see, without enzyme, reactions will take place, but they'll take very long. So enzymes help to speed up the reaction. And you can see how they speed up is the enzyme catalase, which is found in nearly every living cell. They can break down 40,000 molecules of hydrogen peroxide every second. Now, that's enormous. So catalase enzymes can break down 40,000 molecules of hydrogen peroxide. H2O2 is hydrogen peroxide, and this needs to be broken down. It's very harmful for the cells. So a catalase is present in all living cells, mostly all living cells, can break down 40,000 molecules. But if there was no catalase, still the reaction would maybe take 1 or 2 hours. But here, of course, it is happening every second. Now, factors that affect enzyme activity are number 1, temperature, number 2, pH, number 3, enzyme concentration, and number 4, substrate concentration. Now I give you a very simple example of this is that this is an enzyme and say these are 100 active sites. Now the substrate, you only have 10 of them. Well, then of course the reaction is going to stop as soon as 10 substrates are finished. But then you have 100 substrates. Well, then the reaction will stop. But if you have a lot of substrate, then you see now the active sites will become the limiting factor, because the active sites will now be less and the substrate will be now lining up to fit into the active site and the reaction to take place. So that is when the rate becomes constant of an enzyme reaction. Now coming to naming enzymes. Now it's very easy. I mean, it's a rather a weird phenomena that whatever it works on, like if it works on lipids, then it's going to be called lipase. So there is an ASE at the end. I like this meme about it. Protein, protease, peptide, peptidase, maltose, maltase, sucrose, sucrase. So basically, naming an enzyme is easy. If you don't remember it, whatever it's going to work on, just write an ASE at the end of it. Now let's study the effect of temperature on enzyme action. So here you have the percent maximum activity on this side, and you have the temperature on this side, and you can see it's in degrees Celsius. So as you can see from here to here, this is the temperature is increasing, so kinetic energy is increasing. And here enzyme activity doubles for every increase in 10 degrees Celsius. So if you look at 10 degrees and at 20 degrees, the rate will have doubled. Collisions are happening more frequently due to faster molecular motion. More the kinetic energy, more the temperature, more the effective collision. This is a word which has been asked to be used in your syllabus. More the effective collisions. Now, as it reaches the optimum, and after the optimum, you see there's a decline. The rate is decreasing now. And this is why, because at high temperatures, I don't agree with this, at higher temperature above the optimum, the enzyme are denatured, means that it's not complete denaturation. That is why we call this partial denaturation. Because still the reaction is taking place, but it is slowing down. So this is called partial denaturation, and the rate is decreasing. Why? Because the heat breaks the bonds and causes the shape of the enzyme and the active site to change, so the substrate no longer fits. And finally, when it reaches the zero, that is when all the active sites have lost their shape. And they can usually not be reversed. Usually, if you decrease the temperature, the shape of the active site cannot be regained. So as we increase the temperature above the optimum, the rate is decreasing because effective collisions are decreasing. The word which we are asked to use now is effective collisions are decreasing. Now another graph showing the same optimum temperature of enzyme, and you're saying that the temperature increases in activity, that denaturation decreases activity, and how the rate of reaction is on this side, and you have the percentage activity of the enzyme on this side. So optimum temperature of the enzyme, and you can see here it is between 10 to 47, and then after that it's starting to decrease. Now, how can we study the progress of enzyme catalyzed reactions? How can we study the progress of a student? We can do a test in the beginning, and then we can do a test every month to see how your progress is going in biology. I know you're not going to like that, but I think a monthly test would help me to see your progress. Now, in an enzyme catalyzed reaction, you can see the enzyme amylase breaks down starch into maltose. Now, starch was initially the 1000 molecules, and now how much time is it going to take to become zero? So that means all the starch has been used up. Or you can measure the concentration of the product. So originally the concentration was zero, and then after how much time? Did it take 1 minute? Did it take 90 seconds? To finally reach 10,000 maltose molecules. So either we can study this is one situation, which is measure the concentration of the reactants. So starch is the reactant and is being converted to maltose, or measure the concentration of the product or the products. So either we can measure how much time it takes from the starch to completely be zero and all the starch is being used up, or we can start measuring maltose, and then finally a level will come, but after that it won't increase because all the starch has been converted to maltose. You have to understand is that a time will come when the maltose will be first 50, then 100, then 200, then 300, then 400, and finally it will reach 10,000. Why is it am I saying not more than 10,000? Because all the starch has been converted to maltose. No more starch is left. Unless, of course, all the starch is converted. So we can measure the time taken for this final. Okay, how much time did it take for the maltose? So the maltose will be the final. Okay, after that the maltose is now not increasing. So that is the end. All the starch has been converted to maltose. So please understand, how do we study progress of enzyme control catalyzed reactions? Number one, measure the concentration of the reactant. So we measure, okay, starch present? Yes, still present. Okay, every 30 seconds we take a sample. And then finally we say, okay, no more starch is present. Okay, how much time did it take? 90 seconds. Okay, so at 90 seconds, all the starch was converted to maltose. Now, every enzyme has its own optimum pH as well. Just like enzymes have an optimum temperature, they have an optimum pH as well. And the loss of activity is only truly denaturation at extreme pH, since between optimum and these extremes, the loss of activity is reversible. You see, so in denaturation by temperature, you could not reverse it, but pH, if you alter the pH, then it is reversible. Now, enzymes are most optimal or active at optimum pH because they contain R groups of amino acid with proper charges at the optimum pH, and they lose activity in low or high pH as the tertiary structure is disrupted. Because you see, protein molecules have hydrogen bonds and ionic bonds and disulfide bonds. So the ionic bonds would be affected by pH. pH is power of hydrogen. So the ionic bonds would be affected, and this would, of course, make them lose their shape. But then that's a reversible because as soon as you make the pH the same, the optimum back to the optimum, it will start functioning as well. Now you can see here how the different enzymes, library amylase, pepsin, and alkaline phosphatase, all are working at different pH. So this is working at 21.5, library amylase is working at 6, and alkaline phosphatase is working at 9. So different enzymes working at different optimum pH, and the rate of reaction for them is the fastest at different pH. So here is the pH range, and here you can see all these different enzymes working at. Pepsin is, of course, the most interesting enzyme which works in your stomach, because when you eat food, the food contains a lot of bacteria or maybe viruses or anything. So the acid and the pepsin work very well together to destroy those disease-causing microorganisms, which are called pathogens. Now, looking at the two categories of enzymes, the first one is intracellular enzymes. Intracellular enzymes are those which are produced in the cell and they will work inside the cell. So, this is the cell and they will work inside the cell. They will not be thrown out of the cell. So these are reactions that take place within a cell. It could be in the cytoplasm or within an individual organelle, which is inside the mitochondria. So enzyme involved in aerobic reaction, respiration, which organelle would this occur, would be in the mitochondria. Enzyme that modify and process lipids, which organelle would this occur, is in the smooth ER. Some of these intracellular enzyme reactions take part in a metabolic pathway. So the aerobic respiration, which takes place inside the mitochondria, that would be the intracellular. Intra means inside a cell. But there are some enzymes which are also extracellular. So the extracellular enzymes will say this is a cell. So this enzyme is made in the cell, but then it has to be shipped out of the cell. Like for instance, if you remember in the stomach, in the wall of the stomach there are cells and they make this enzyme pepsin, and then the pepsin is thrown into the stomach so that it can digest the chicken kadai that you had, or the omelette that you had, or the fish that you had. So it is an extracellular enzyme, but it's made inside the cell, but it doesn't work inside the cell. This pepsin would be, of course, dangerous inside a cell because it would digest all the enzymes present in the cytoplasm. So intracellular enzymes are synthesized and retained in the cell for the use of the cell itself. They are found in the cytoplasm, nucleus, mitochondria, and chloroplast. Example: oxidoreductase, catalyzed biological oxidation, and enzymes involved in reduction in the mitochondria. Extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. An example they've given you is digestive enzyme produced by the pancreas, like the trypsin, the iripisin, and the pancreatic amylase. They are not used by the cells in the pancreas but are transported to the duodenum. So this is the pancreas, and then they are this tube which opens in the duodenum. And the cells of the pancreas make certain enzymes, but they are then thrown into this duct, and then they come here and work here. So they will be produced here, they will be produced in the cells, but they will enter this pancreatic duct and they will be brought here and they'll be mixed with the food in the duodenum, and they will digest the food in the duodenum which you've eaten and which remained in the stomach for 3 hours but now is entering the duodenum, and it will be digested by the three enzymes produced by the pancreas. So intracellular enzymes and extracellular enzymes. Now it's very interesting to see the different microorganisms which have been discovered and how they have different optimum temperatures. And the interesting is, of course, this is the spoilage microbes. Spoilage microbes means the ones that is, decomposition. Then the pathogenic microbes, they're the same as, the ones which cause infections of the human body, typhoid, cholera. And then we have the thermophiles. Now the thermophiles are the ones which are very interesting because we can use them in a number of industrial processes in which we don't want to put in new enzymes. So these thermophiles, we've extracted enzymes from them and then we use them in PCR, polymerase chain reaction. Now you can see their optimum is 60 degrees. And then we have the hot spring volcano microbes. Oh my God, I mean, look at that. They are something like 90 plus the optimum. So they would be denatured at 110. So it's not very easy to kill them. So you can see how these are the growth rates of different type of microorganisms in response to temperature. So the normal question that we often ask you is, what is the optimum temperature of enzymes? Well, you cannot say anything because if I have not specified, yes, if it's a human enzyme, yes, it works best at 37. But if I just said optimum temperature of enzymes, then I'm afraid you can't give an answer because these are these microbes which are working best at 95 degrees Celsius. So this is what I want you to understand is that this discovery has of course helped us to use these enzymes for a number of processes which we use in genetic engineering and other processes. So it is not that all enzymes work best at 37. It's only the human enzyme that work best at 37. So different enzymes will have different optimum temperatures, and this is something which you all must know. And you must be able to do the MCQ on it. Now, I'm going to end this video here. Thank you for watching, and I will continue with the practicals in the next video. So thank you very much for watching, and I hope this is helpful to you, and best of luck. Also, as I end this video, I would like you to say this prayer whenever you have the time. This is a very good prayer. And O Lord, give us in this world that which is good, and in the hereafter that which is good, and protect us from the punishment of the fire. So please repeat this. Maybe you know the Arabic part of it. If you don't know the Arabic, you can even say the dua in English. And I'm sure this is going to be helpful to you in your studies and in your future.